by the chemicals that are associated with it. All right. So you're trying to, you're playing Army, whatever, and you've got a, or
playing Army,
Indians and cowboys, and so you're trying to climb the stockade, and the opposite team has coated it with. No. That's a gross picture. I was trying to think of the word lard.
There's a whole lot of them out there.
To make it really slippery. So that would, the stockade wall itself would be the mechanical barrier, and the lard and saliva would be the chemical barrier, all right? But first, they're both preventing you from getting inside the stockade, getting inside the fort. So that's the first line of defense. The second line of defense is both cellular and molecular. So once you're actually inside the body tissue, so even just because it's in the stomach or it's in the small intestine doesn't, you're not really truly within the body. Okay, you haven't crossed the plasma membrane of the cells lining that organ. If you do, you're a pathogen and you make your way between the epithelial tissue or the integrity has been opened via an ulcer or something, then we start a series of events that I call the second line of defense. And so the first process, we talked about phagocytosis earlier, and these are the major cells of the white blood cell system that function via phagocytosis. Our B lymphocytes can also do it, but that's not their major mechanism of defense. And so this is showing a macrophage ingesting bacteria. And remember, of course, our macrophages are mature monocytes that have increased their population of lysosomes. And the lysosomes contain the proteases and break down the wall of the viral or bacterial component. Now, a macrophage, remember, is also an antigen-presenting cell. So on Thursday, we're going to talk about that process of antigen presenting involving our lymphocytes. But just as a phagocytic cell, macrophages, neutrophils are important in removing the foreign tissue. Now this is a bit of a conundrum. Natural killer cells are lymphocytes. As a lymphocyte, it has receptors on its surface. But these receptors are not recognizing an antigen. They're recognizing the non-specific region of the antibody. So remember we talked about basophils, and a basophil will come in contact with the constant region of IgG. So these are IgEs. We'll talk about these on Friday. And a basophil coming in contact with that releases mast cell. Releases histidine. So when we look at an antibody, which we'll also do late Thursday or Friday, this is the variable region, this is the specific area of an antibody, and this is the constant region. So this will identify whether it's an IgE or an IgD or an IgG. And so thousands of different antigens can be recognized by this antibody, but this region here, by antibodies similar to this, but this region here will stay constant.